Cancer has been a big focus in the scientific world for years now because survival rates are very low. Scientists everywhere have been working to figure out what exactly triggers cancer in patients and what allows it to grow and spread. Over the years, many theories have come up, but one of the most interesting and convincing ideas is the concept of cancer stem cells or CSCs. A promising solution to this problem is CAR T-cell therapy. Let's have a quick look at what exactly they are. T cells. They are a type of white blood cells. The bone marrow produces hematopoietic stem cells from which we get the myeloid progenitor cells, which produces the RBCs, platelets and other white blood cells, whereas we have the lymphoid progenitor cell which produces the T lymphocyte and the B lymphocyte. The lymphoid progenitor cell then makes a pre-T cell which ultimately develops in the thymus, which is a gland located in the chest. Once mature, they circulate in the bloodstream and lymphatic system, searching for foreign invaders like viruses, bacteria and cancer cells, and they can mount an immune attack against them. T cells and their receptors. The T cells have their own surface proteins called receptors. These receptors can recognize cells that have abnormal antigens. T cells act as a surveillance system for abnormal cells, becoming active when a receptor recognizes an abnormal cell. The activated T cell goes to work destroying the abnormal cells and activating other parts of your immune system to help find and destroy abnormal cells. But the T cell receptor don't always detect the cancerous cells. This is where CAR T cells or Chimeric Antigen Receptor T cells come into picture. They are specially engineered in the laboratory and are made to recognize a specific antigen on the surface of cancer cells. In the lab, the collected T cells from a patient are genetically modified to produce special receptors called chimeric antigen receptors on their surface, or commonly called as CARs. These receptors, they are synthetic molecules. They don't exist naturally. A CAR is a molecule that combines an antibody fragment that can recognize a specific antigen on the cancer cells with a T cell signaling domain that can activate the T cell upon binding with the antigen. The primary components of the CAR structure include the extracellular domain, which recognizes antigen, the intracellular domain, which contains a CD3 signaling domain and one or more co-stimulatory molecules, and finally the transmembrane domain, which connects the two previous parts. A growing number of CAR T cell therapies are being developed and tested in clinical studies. Although there are important differences between each specific therapy that can affect how they function in patients, they all have similar components. Each CAR bridges the cell membrane. Part of the receptor is located outside the cell and part within the cell. The part of the CAR that extends out from the cell surface is usually composed of fragments or domains of lab-made antibodies. Which domains are used affects how well the receptor recognizes or binds to the antigen or tumor cell. The internal part of each CAR has signaling and co-stimulatory domains. These transmit signals into the cell after the receptor interacts with an antigen. The different domains that are used can affect the cell's overall function. One of the primary functions of CAR T cells is to destroy cancer cells. Once the CAR T cells are injected into the bloodstream, the CAR T cell receptors detect and destroy the cancer cells. Thus, CAR T cell therapy is a type of immunotherapy that uses the genetically engineered T cells to fight the cancer cells. This type of therapy uses the power of patient's own system to fight certain types of cancer, such as blood cancer, including lymphomas and certain kind of leukemia, and most recently even multiple myeloma. Several approved CAR T cell therapies work by binding to CD19 antigen on cancer cells and killing them. CAR T cell therapy gets the nickname living drug for a few reasons. As it is made from living cells, unlike traditional medications that are chemicals or compounds, CAR T cell therapy uses a patient's own immune cells, specifically the T cells. Because they're living cells, they can grow, multiply, and continue fighting cancer cells for an extended period of time. So what are the steps that CAR T cells undergo from the onset of infection till reaching the tumor site? The first step is trafficking. After injection, the CAR T cells start circulating in the patient's bloodstream till reaching the tumor site. Second step is infiltration. After CAR T cells arrival, they have to enter the tumor cells, and only in solid tumors they have to overcome the tumor microenvironment that promotes the growth of the tumor by supplying it with nutrients through special blood cells. It also secretes cytokines and repel immune cells away. This gives the tumor microenvironment its immunosuppressive property. CAR T cell therapy has been also recognized as personal medicine, as the therapy is customized for each patient. Engineering the T cells to recognize and attack a specific marker on the patient's specific type of cancer cell gives it the personalized approach. This personalization also increases the effectiveness of the treatment. So how is CAR T cell therapy used to treat cancer? Healthcare providers collect blood to obtain the T cells, from which the T cells are separated and removed, and the rest of the blood is returned to the patient. The T cells are then genetically altered to have the special receptors called as CARs, which are designed in such a way to recognize specific proteins or antigens found on the surface of cancer cells. Millions of CAR T cells are grown like this, and after genetic modification, the CAR T cells are infused back into the patient's bloodstream. The modified CAR T cells now have receptors that allow them to specifically bind to the patient's cancer cells. Once attached to cancer cells, the CAR T cell activates the immune response, and they release signals that attract other immune cells and initiate a powerful attack against the cancer. These modified T cells are then able to multiply and kill the cancer cells throughout the body. What are the core strategies and innovations in CAR T cell therapies? First is the synthetic biology and engineering. It enhances the CAR T cell targeting, specificity and persistence by integrating switchable adapters, dual antigen recognition, TCR CAR hybrid features, and engineered secretory cytokines. Second is the manufacturing advancements. Improving CAR T cell functionality and delivery through CRISPR-based gene editing, optimizing the viral and non-viral vectors, and selecting specific T cell subsets for long-term persistence. Targeting novel antigens. Expanding CAR T cell applicability by identifying tumor-specific neoantigens, targeting the cancer stem cells, and engineering CARs to eliminate resistant cancer populations. Enhancing solid tumor targeting. Improving the CAR T cell infiltration and survival in tumors using chemokine receptors, matrix degrading enzymes, and engineered armored CARs to counteract the immunosuppressive environments. Synergistic combination therapies. Enhancing CAR T efficacy by combining with checkpoint inhibitors, kinase inhibitors, radiation, and chemotherapy for stronger, more durable anti-tumor responses. Allogenic CAR T cell therapy. Developing standardized, ready-to-use CAR T treatments using healthy donor T cells and genetic modifications to prevent immune rejection. And lastly, advanced strategies to mitigate toxicities. Reducing toxic effects by integrating safety switches, localized CAR T delivery, predicting screening, and prophylactic medications to minimize severe immune responses. Clinical trials of CAR T cell therapy. The first clinical trial of CAR T cell therapy was conducted in the early 2000s. These trials showed that CAR T cell therapy was safe and could produce some positive results in patients with cancer. However, it was not until the late 2010s that CAR T cell therapy became a widely used treatment for cancer. Clinical trials conducted globally have shown promising results in end-stage patients, especially in patients suffering from acute lymphocytic leukemia. CAR T cell holds immense promise for the future of cancer treatment because of a few advantages. First is high emission rates. CAR T cells have shown success in treating certain cancers, particularly blood cancers like leukemia and lymphoma. Studies have shown remission rates as high as 80% in some cases, exceeding what traditional therapies can achieve. Personalized approach. Unlike chemotherapy or radiation that target all rapidly dividing cells, CAR T cells are programmed to go after a specific antigen or cancer cells. This personalization minimizes harm to healthy tissues and reduces the side effects. And lastly, living drug. As mentioned earlier, CAR T cells are engineered to multiply and persist within the body. This means that they can offer long-term protection against cancer recurrence. They act like a living defense system, continuously patrolling for and eliminating the cancer cells. But what are the challenges to address? Manufacturing complexity. Creating CAR T cells is a complex process that can be expensive and time-consuming. Researchers are working on streamlining the manufacturing process to make it more accessible. Solid tumor applications. CAR T cell therapy has shown great results in blood cancers like leukemia and lymphoma. However, solid tumors pose a different challenge. Solid tumors often lack ideal targets or antigens for CAR T cells, and the tumor microenvironment can suppress the activity of those engineered cells. Side effects. CAR T cell therapy can cause severe side effects like cytokine release syndrome or CRS, where the immune system goes into overdrive. Managing these side effects is crucial for patient safety. Another concern is off-target effects where CAR T cells attack healthy cells due to targeting the wrong antigen. Research is ongoing to improve the safety profile of this therapy. Durability and persistence. While some patients experience long-term remission, CAR T cells can sometimes become inactive or ineffective over time. This can lead to relapse. Researchers are looking at ways to enhance the persistence and memory function of these cells. Overcoming tumor escape. Cancer cells can mutate and lose the targeted antigen, rendering CAR T cells useless. Developing strategies to target multiple antigens or using smart CAR T cells that can adapt to these changes are areas of active research. Not cost effective. The process of developing this personalized therapy makes the cost of treatment very high. It is beyond reach for the common man to avail this treatment. Undoubtedly, with CAR T cell therapy, an exciting new era in cancer research awaits. Researchers around the world are constantly working on new ways to develop new and improved CAR T cell therapies. Optimizing CAR T cell design and delivery would lead to cures for more malignancies. They are also working to make CAR T cell therapy more affordable and accessible to more patients. They are also exploring a way to work around allogenic CAR T cells derived from healthy donors along the traditional CAR T cell therapy using a patient's own cells. Research developments like these can potentially bring down the cost of the treatment, making it more accessible and affordable. As research and development continues, there's definitely hope that CAR T cell therapy will become the most recommended treatment option, improving outcomes at large. Thank you.